My name is Seung Kim from Chongbuk National University. I'm very honored to chair this morning session. Due to time schedule, let's get started with the first speaker, Professor Park Kwang-sung. Please refer to the details in the book. Okay, please.
Yeah, thank you for your kind introduction. I'm sorry, because the plenary session was on the other side, and I thought after plenary session, my talk will be started. I'm Kwang-sung Park from Jeonnam National University. I'm very happy to share my data from Korean centenarian study. Nothing to disclose. As you know, the WHO declared the UN Decades of Healthy Aging in 2021. And then we are middle in 10 years of healthy aging strategy. And then the worldwide increase in life expectancy and rapid aging, the population have increased medical interest in centenarians, but we have limited data regarding to regional characteristics in Korea. As you know, the population pyramid in South Korea, in 2000, the population was bell-shaped, peaking in 30s and 40s, and in 2020, became base-shaped, and the peak shifting to 50 to 60. And then what are the trends and the projection of elderly population in Korea? In 2000, the Korea became aging society, and 2020, entered the aged society, and now in 2025, this year, become a super-aged society. And how about the life expectancy and the gender difference? And in 2000, the female life expectancy was 79.7 years, and the man is 72.3. So the gap is 7.3 years. And currently, the life expectancy in women is 86 years. And man is 80 years, and still gap is a little shorter than 5.8 years. How about the major changes, causes of death from disease among all the adults in Korea? In 2000, the stroke read first in 23.7%, and then followed malignant neoplasm and ischemic heart disease. In 2019, the first rank is malignant neoplasm, 26%, and followed by lower respiratory tract infection and ischemic heart disease. And then the stroke is a big change from 2000, and maybe influenced by the emergency care system in from the government. There's a big decrease of the stroke. About the centenarian in Korea, in 2000, the total number is around 934, and then in 2022 is increased to 1,400. My talk will be divided in three parts, and the first part is designation of Gugok-sundam, Korean longevity belt. As you know, the Professor Sang-cheol Park published the first comprehensive study on human longevity in Korea in 2001, and then designate the longevity belt, and based on areas with a high proportion of people over 100 years and over 6% of longevity rate, and selected Gure, Gokseong, Suncheon, Damyang, and so it's called the Gugok-sundam. And where is the Gugok-sundam? We are here in Gwochang, and Gwochang is near Gwangju, and then the Damyang, Suncheon, Gokseong, Gure is located here around the Mount Jiri, the big mountain in Korea. And then he concluded that some gene influenced longevity and in gender, the female dominance and the advantage of the traditional Korean food, and then the local culture and tradition influence, and then mountain impact and geographic variables. And actually, I'm a urologist as a surgeon, and then why I could dive into the Korean longevity study, and because I met Professor Sang-Chil Park in 1918. And then we did the first study from Jeonnam National University, the Korean centenarian study. And we measured the characteristics and the physical and mental health and centenarians and the interaction with family and the community. And then we published a book of the Korean centenarians, 20 years changed, and then my talk will be based on this book. And 2001 and 3, the male to female ratio is 1 to 13 and decreased to 1 to 5 in 2018. And how about the disease, chronic disease history? In 2003, very rare prevalence of diabetes, hypertension in centenarians, and it increased to diabetes to 5.6 and hypertension 40.4%. Still very rare prevalence compared to general population. And how about the subject health status? You can see the excellent good health status in 2/3 of the centenarians. And then about the sleep duration is around 9 hours and 8 hours. So if you want to be a centenarian, please sleep very well every day. And about the alcohol consumption and smoking rate, still no smoking experience is 66% or 75%. And so actually the smoking rate and alcohol consumption rate decreased overall in 2018 compared to 2001. And how about the range of daily activity? And within the room, it's 37% 4 point in 2001, and then in 2018 it's decreased to 20.2. And then the activity duration, range of activity duration in 2018 is 7 out of 10 centenarians were active over 30 minutes in 2018 study. About the ADL score, and then interestingly, there was very little decrease compared to 2001. And then we are curious about why the ADL score is different from the other data. And then we found that it's possibly due to 20% of centenarians live in care facilities. I'll show the other slide. And then the cognitive ability we measured by KMMSE, and then it's similar between 2001 and 18. And then women's cognitive function is lower than men. It's a well-known data. How about the depression scale? Score was lower in 2018, and it means more better depressive score compared to 2001, but still in 2001 it's 8.3, so mild cognitive depressive score. And then how about the lifestyle? You can see the photo of 2001, and then most centenarians wear the white cloth. It's very popular in color in Korea. But in 2018, you can see the color of the clothes, and then it's a big difference of the lifestyle or cloth style. And you can see the Professor Jung-Hwa was a graduate student, and he participated in the 2001 Korean study, and now he's a professor of Jeonnam National University. And who live they with? And then in 2001, most centenarians lived with family, and then only 12% live alone. But the big difference is it increased to 32% live alone, and 17% live in care facilities. So it's a big change. And then 2022, we did the Korean centenarian study, even though we are still living in a COVID-19 pandemic period. At the time, we wear the facial mask and then did the daily COVID test. And then the difference of the study compared to the previous study, we measured the visual acuity and hearing. And let me show a little bit data. And then visual acuity is around 0.2 to 3. This range, okay, large number they can see. But the problem is only 8% of centenarian wore the glasses. About the hearing, and they, interview, they show that the daily activity limitation due to hearing was 71%, but only 12% of centenarian were the hearing aids. And then we did a Hwasun centenarian study 2023, and then Gwangju study in 2023 and then 2024. And then we published another book in English version, Secret and Evolving Trend of Korean Longevity. And then I give for the foreign speakers this book today. And then we established the Korean Centenarian Study Group in Jeonnam National University, and Professor Sang-chul Park donated his books and reports to this laboratory. And then finally, the Health Joseon newspaper summarized our whole study. And then, for example, to be a centenarian, quit smoking and drinking alcohol, and then should be independent and active and good, well sleep, and have some family history, and then very good social network, and then stay the good lifestyle. And then in this early June, I attended the International Centenarian Consortium meeting hosted by the New England Centenarian Study and Boston University, and which was held in Cape Cod, Massachusetts. And it was a 29th meeting, and Professor Leonard Poon and Peter Marin and attended. And then actually the hosting is Dr. Thomas Paul, and then we had a very good meeting. And the next year, 26th ICC meeting will be held in Korea, and then we will celebrate the 30th anniversary of ICC. Please join the next Korean meeting. And then thanks all the participants of the Korean Centenarian Study, and then thank all. I am sorry that I cannot introduce all of our members, and then I thanks all the members of Korean Centenarian Study Group. Thank you very much for your attention.
Thank you. Let me introduce the second speaker, Dr. Kim. Please refer to the book in details.
Hello, my name is Yuri Kim. I'm the EP cardiologist to doctors for the arrhythmia patient. Actually, before I start my presentation, I am sorry to apologize to everyone because most of my slide is in Korean, because the organizing committee didn't tell me the speaking is have to be in English. So yeah, I will talk about this. And as the previous speaker says, in Korea, the average age is increasing. Maybe in this year, the female average age is over 90 years. And now, in southern part of the Korea and most of the Japan are already the super aged society. So in the heart, we gotta have this function. The heart has like a pump to go. The blood could go through the whole body. So in the arrhythmia means there is some problem in the heart to running the heart coordinately. Here is the diagram for the heart. And the normal heart, we started the SA node at the first sign of the heart, and then to go to the AV node and both ventricles, like this. So in arrhythmia says, in Korean, we call it the 부정맥, means there is the 정 means not the normal means is the regular things. So the arrhythmia is not the regular heartbeat. So the AFib is the most common arrhythmia disease we have to treat. So do you know why the AFib have to be treatable? Because the AFib makes the mortality higher than the normal person and 5 times more stroke and without AFib. In the aged patient has more AFib. Usually the 2% of the population has the AFib and under aged 60 years. But over the age of 80 years, the percentage of the prevalence is more than 8%. So 1 in 10 has an AFib. And now we treated the AFib coordinately, but still have the struggle with the ischemic stroke. And also, there is some complication is more with according to the increasing age. So how we can diagnose the AFib? We do the EKG. And there is some problem. It's hard to diagnose the arrhythmia. It's because the symptom is different, and also the symptom has the irregular and the paroxysmal. And there is a patient has no symptom with a permanent AFib. And so we have to the EKG with the patient has a symptom. But you know that the EKG is hard to do in your home and your daily routine. So there is another tools. There's 2 kinds of the wearable device using the ECG and also the PPG. The EKG, as you all know that. And the PPG is the single device to detect the heart rate. Use the lead. And here is the trends of the wearable device. And now in Korean, 1/4 population has the wearable device. And to detection of the arrhythmia, there is 2 kinds of the wearable devices using the ECG and the PPG. The one is the continuous monitoring, and the other is the intermittent monitoring. And for the continuous monitoring, there is a big study, the Apple Heart study. The inclusion is the people who bought the Apple Watch and the 400,000 population enrolled this study. It's more than any other study in the clinical study. With the result, only the 0.5% has the irregular heartbeat alarm with the Apple Watch. But you know, over the 65 years, there is a percentage of the irregular heartbeat is 3 point. But under the 40 years, only the 0.2. So as you know that the older person has the more AFib and they get the more irregular heartbeat alarm. And with the PPG alarm, the positive predictive value is that someone has the irregular heartbeat and then diagnosis the AFib is 84%. So PPG smartwatch can detectable the AFib for the screening. And there is another device, the ring type, in Korea. This device can take the blood pressure also with the PPG data and also the ECG data. And we have another tool to detect arrhythmia. The arrhythmia has, as I said before, the paroxysmal, so intermittent ECG check like this, the ring device and also the plate with the smartphone. With the plate with the smartphone, there is a more AFib diagnosis than the routine checkup. And also the smartwatch with the ECG, the Galaxy Watch and the Apple Watch can detect the ECG with the contact your finger to the ECG. It's different from the PPG data. You have to contact your finger to the smartwatch. And here is my patient, the 68-year-old female. She got an intermittent palpitation and the skip beat. We do the event record in the outpatient clinic for 2 weeks. He got a 140 event recorded by her, but there is no significant arrhythmia in that time. But after he bought the Apple Watch and bring the data, here is the PVC, the very frequent PVC with the ECG. So this is the wearable device can detect and diagnose the arrhythmia. And there is another tool, the Holter monitoring, the ambulatory ECG monitoring. Here is the Holter, Dr. Holter. He invented the Holter device in late 19th. And now we do the single lead wearable ECG Holter for not only the 24 hours, but also the 7 days and 2 weeks. There is a two type, the lead type and the patch type. The first time is the lead type. We compared the 24 hours conventional Holter and also 27 the wearable lead type ECG, the same time. Interestingly, the conventional Holter usually used in only the 24 hours. There is the detectable arrhythmia is only the 10%. But after the 3 days wearable ECG, the increase the detection rate of the arrhythmia to go up to the 20%. And this is my own experience. Can take the wearable device for 48 hours, the lead type. The first day, I have no symptom, and also here is the very good recording. And well, I'm healthy now, so I have no arrhythmia yet. But there is some problem to lead type ECG, because in the running, the noise problem is like this, and also the detachment. So we have to reinforce the lid to attach the skin. And after that, there is another wearable ECG, like a patch type, to overcome the lid problem. And compare the telemetry data and this wearable patch type ECG, very good result. Even the ICU patient, the wearable ECG can detect more than the telemetry, the conventional telemetry. And here's my patient. It's 85 years female and intermittent palpitation. We applied the patch type ECG, and he get a day 4. There is some irregular heartbeat, and revealed the atrial fibrillation. So with the conventional Holter, we applied only under the 48 hours. So without this patch type ECG, we cannot diagnose this patient to the AFib. So this is the advantage to use the wearable ECG. And also here's my own experience, the wearable ECG for 98 hours. First 2 days, I have nothing to feel or disturb it. But after 3 days, I have some skin problem, and we have to overcome that kinds of the disadvantage to use the patch type ECG. And the more ECG we get, we need the more analysis we have to. So now we apply the AI to analyze the wearable ECG data for detect the arrhythmia. Here is the detection rate. It's over the 95% already. But there is some problem to use the wearable ECG for everyone, because there is only the single lead ECG, not the multiple channel ECG compared with the conventional Holter. So here is some problems. And also the privacy had another problem too. And after we detected the arrhythmia and AFib, there is another problem too. The aging is for the not only for the strokes, but also for the bleeding risk. But the true value of the true positive effect is for the anticoagulation is going up to the patient with the age. And so the underdosing is not for the benefit for everybody. And there is another problem too. The elderly patient has had the multiple drugs. The polypharmacy is another problem too. And here's the summary. The wearable ECG is for the very usable tool to detect arrhythmia in patient with the age. And also, this continuous monitoring is how to the people to detect the arrhythmia in daily life, and the low cost. But there is some problem, is the privacy, and also the analysis is to the ECG data is the more we get the ECG, and it's hard, the harder we have to analysis more. So here is another problem. But after we developed the technology, we overcome these kinds of the disadvantage. Thank you for the listening.
Thank you, Professor Kim. Let's move to the third speaker, Professor Park from Arju University School of Medicine. He will present the data, functional rejuvenation of the elderly organs. Yeah, please.
Hey, thank you, Chairman. Today, I wanted to share our idea and the data with the audience about the aging process. We researched on organ aging process in human, specifically focused on the skin. Today, I present two subjects. Firstly, when studying aging, it is important to distinguish between pathologic condition and non-pathologic condition. Secondly, in non-pathologic condition, the fully senescent cells do not constitute a large proportion. Before my presentation, I wanted to ask one question. Do you believe that the senescent cell percentage is increased with aging? Most of the scientists believe that the senescent cell is accumulated in old-aged tissue. Therefore, they wanted to target the senescent cell to overcome the aging. In the aging study, the epithelial cell differentiated from stem cell and rapidly renewed. For example, in the case of epidermal keratinocytes are replaced within 1 month, and also gastrointestinal tract epithelial cells are replaced with a new cell within 1 week. Therefore, there is not enough time to undergo senescence. However, particularly in fibroblast or melanocyte, there is a possibility of accumulation within the tissue and sufficient time to enter the senescence process. Therefore, to study aging process in skin, we should focus on fibroblast and melanocyte rather than keratinocyte. One more thing, I already mentioned that we should to consider pathologic condition and non-pathologic condition skin separately. This is because the senescence mechanism in exposed skin is triggered by UV radiation or chemical irritation. But non-exposed skin is induced by replication. We found that the melanocyte and the fibroblast become to senescent cell in pathologic condition skin. I show the evidence in following slide. For example, the senescent fibroblast accumulated in hyperpigmented senile lentigo. As a Korean language, the 노인성 흑색 반점. It can be among the patient. Approximately 10% of fibroblasts show senescent phenotype. As another piece of evidence, the accumulation of senescent fibroblasts has also been observed in melasma. As a Korean word, the gimi. We have identified two proteins derived from senescent fibroblasts that can regulate melanogenesis. First is SDF1, which is abundantly expressed in young fibroblasts, but downregulated in senescent fibroblasts. Second is GDF15, which is expressed in lower level in young fibroblasts, but is highly expressed in senescent fibroblasts. As a mechanism level, SDF1 signaling reduced cyclic AMP CREP signaling and suppressed MITF expression, leading to the inhibition of pigmentation. In contrast, GDF15 through the GSK beta-catenin pathway and increased the MITF expression and enhanced the pigmentation. Therefore, senescent fibroblasts show a decrease in SDF1 and an increase in GDF15 resulted in increase in skin pigmentation. We hypothesized that the elimination of the senescent fibroblast would improve skin pigmentation. To eliminate this senescent fibroblast, we performed the ablation of senescent cell using radiofrequency technique. Approximately 6 weeks later, a significant reduction in senescent fibroblast was identified. Over the age of 60 years old, treatment using this method led to improvement of skin pigmentation. In the case of melanocyte, the senescent melanocyte identified in idiopathic gutta hypomelanosis. In contrast to senescent fibroblast, senescent melanocyte reduced skin pigmentation. You can find that P16 positive senescent melanocyte in hypomelanosis region. The melanosome are transported to the peripheral region of the cell by several intracellular motor proteins. For example, LAP27, MYO5A, and MLPH. And then secreted through the exocytosis. However, in senescent melanocyte, the expression of motor proteins is downregulated. This downregulation prevents melanosome moving to the peripheral area of the cell. As a result, the melanosome are unable to the exocytosis and accumulated within the senescent melanocyte. That we confirmed it using electron microscope. Until now, I explained in pathologic condition and the senescent fibroblast and the melanocyte play an important role in skin aging process. However, how about in normal skin aging process? In many studies, including our data, it has been observed that the fully senescent fibroblast and the melanocyte do not increase in old age skin tissue. I show the evidence. It is old-aged mouse RNA sequencing data. Compared with young mouse, old mouse show that senescent cells are increased in some organ, including liver and the tongue. However, in the case of aorta and bladder, the senescent cells were downregulated, and there is no difference in most of organs. In the case of skin, very small percentage of senescent cells were increased, but not significant. A senescent cell is the immune cell, not keratinocyte or fibroblast. This is old-aged mouse single-cell analysis, and you can see the immune cell senescence, not fibroblast and keratinocyte. This is also human skin data from the old-aged human skin. When subjected to single-cell analysis and compared to young skin, there is no increase in P16 expression in keratinocyte and fibroblast. However, we observed the increasing of P16 expression in immune cell, specifically T cell. This data also indicate that the increased P16 expression in old-aged tissue originated from the immune cell rather than keratinocyte or fibroblast. As a conclusion, we found that fully senescent cells were not increased in old-aged normal skin tissue. It is very important because if fully senescent cells are rich in old-age tissue, using the senolytic drug is a good strategy. However, if fully senescent cells do not exist in old-age tissue, the strategy should be changed. Based on our research, we can draw the following conclusion. Senescent cells do not increase in aged normal tissue. However, this conclusion does not mean that it is a young cell. Therefore, we conclude that the aged tissue cell is neither fully senescent nor young cell. This means that intermediate stage cell exists in old aged tissue. We referred it mid-old status cell. In order to identify the existence of mid-old cell in the tissue, we attempt to identify the mid-old cell marker. To find the mid-old cell marker, we used three different approaches. First, we utilized the in vitro culture model. Second, we isolated the stromal region from the aged tissue through the microdissection method, and then RNA sequencing data. Lastly, we utilized the deposit RNA sequencing data. By integrating these three data sets, we derived the following mid-old cell marker. Young cell was negative for P16 and P21, but positive for CXCL12 and SLIT2. The mid-old cell was negative for P16, but positive for P21 and serum amyloid A1. Additionally, they were negative for CXCL12 and SLIT2. The fully senescent cells were positive for P16, P21, interleukin 6, interleukin 8, but negative for CXCL12 and SLIT2. Therefore, potential mid-old cell marker can be considered as SA1 positive, P21 positive, while SLIT2 and CXCL12 are negative. In the case of old-aged mouse, also showed marked increase in SA1 expression. You can find that the SA1 expression in the skin fibroblasts. Furthermore, serum SA1 concentration also markedly increased in old-aged mouse. Next, we analyzed the young cell markers, such as CXCL12 and SLIT2. The expression was increased in young-aged skin, but markedly reduced in old-aged skin. Therefore, we can draw such a conclusion. In the old-age tissue, the cell is not young cell status. However, it also is not old cell. The cell are mid-old status. So what is the difference between mid-old cell and the senescent cell? The most important thing is responsiveness to the external stimuli. The fully senescent cells do not respond to the external stimuli such as EGF. However, mid-old cell, similar to the young cell, show that significant signaling is transmitted to the nucleus in response to external stimuli. The left is a young cell, and the middle is a mid-old cell, and the right is a senescent cell. When stimulated with EGF, if the color of the nucleus changes, it is interpreted as a response by EGF. Young cell and mid-old cell show similar response. However, senescent cells show no response at all. This observation is very important because mid-old cell can regain cellular activity if provided with appropriate stimulation. The aging process does not continuously increase like a linear function. It is known to have a 3 inflection point around 37 and 60 and 78 years old, where aging accelerates dramatically. But until now, the exact mechanism is unknown. Therefore, we made the following hypothesis. The certain factor prevent aging process until they reach their limit. If over the limitation, the aging process rapidly occurred, we thought that this anti-aging factor originated from young cell. Therefore, we made effort to discover potential rejuvenation factor from young cell. Finally, we identified several kinds of rejuvenation factor, including long non-coding RNAs and proteins. Among them, today I discuss about the SLIT2 protein. As earlier mentioned, SLIT2 protein overexpressed in young tissue, and the expression was downregulated in old-aged tissue. When treating mid-old cell with recombinant SLIT2 protein, we observed a reduction in cell size and an increase in cell proliferation. The mechanism is P21 downregulation, and it shows independent of the P53 pathway. To find the mechanism of P21 downregulation, we conducted RNA sequencing analysis after treating with recombinant SLIT2 protein and found that SLIT2 protein increased the expression of stem cell factor such as SOX2 and OCT4. Furthermore, SLIT2 inhibit NF-kappa signaling pathway, leading to the suppression of inflammation-related gene expression. Lastly, we treated SLIT2 protein to old-aged mouse model. The upper panel represents the control group. You can find that the mouse movement is slow and sometimes show lymph phenotype. In contrast, the lower panel represents the mice treated with SLIT2 protein. As you can see, the mice treated with SLIT2 protein displayed increased activity and movement within the cage. Based on our research, we would like to convey the following point. Senescence cell is a real enemy in pathologic condition. However, the mid-old cell is a real enemy in normal aging process. And the treatment strategy also should approach in different method. I'm 75 years old, and I'm already old and no hope anymore, but it is not true. The cells are not yet aged, and the cell can undergo functional rejuvenation sufficiently. We performed the senescence study in Inflammation Research Center and the Functional Rejuvenation Center, and the France Chanel company also support our research. Thank you for your attention.
Thank you, Professor Park. Okay, the next speaker is Professor Kim, Jung-soo Kim. He will present the data, epigenetic and genomic data across the aging spectrum. Please.
Lifespan. It's a time from birth to death, and organisms begin as a single cell and underwent differentiation and cell divisions and differentiation. And also it interact with the various signals And after certain amount of time, it eventually experience functional decline. So I said it's natural prevail and evolutionary senescence. I'm not going to use the term aging because I think the term aging should be limited, used to the cases where this natural decline is abnormally fast due to accumulated prolonged imbalancing or something. So lifespan, aging hinders a healthy lifespan, and if you consider about the lifespan distribution, then it should be like this. And I want to know whether this information is genomically or genetically determined or not. Probably, if you believe some lifespan is predetermined with the genomic information, but what in real situation, executed lifespan the variance is higher, and we don't usually live our programmed lifespan. So what causes this gap is my interest. And most further, can we extend it is another topic, but I'm not going to talk about these things, but this is also interesting questions. So unfortunately, I'm not going to provide framework of aging, but I'm still gathering evidences from my study. So I provide some fragments what we observed, and I left the organizing framework. I left the assignment to yourself. So to explain about the genetic inheritance, whether the phenotype is genetically determined or not, I brought this example of Huntington's disease. So as you might know, this CAG repeating HTT gene strongly correlates to Huntington's disease. Actually, this is well-known cause of Huntington's disease. And one of my collaborators, Professor Hong Chan-shik, he's dedicated his research on identifying ion channel called TRPC1 and 5 composition might affect the Huntington's disease. But what we don't know is that how these are interconnected. So we just download the public data set and perform some analysis, and we identified enriched calcium signaling gene ontologies are enriched in this data. So maybe this HTT repeats, we just identify HTT enriched individuals and the genes associated with those things, and the functions, we identify the functions are related to this ion channel. So maybe this probably, there are some physical interactions is linking, we are absence of such information, but we also investigate whether those informations are available. And one of my students identified some public data that there are physical interactions of HTT genes and TRPC5. So we need to validate this information, but I believe some phenotypes are strongly determined by genetic factor. So I hypothesize that every phenotype has deterministic background written in genome, and there are controllable different aging or epigenetic factors, accelerated or delayed the phenotype development or not. So I'm talking about the aging gap. So aging differ by individuals, we all know. But aging differ by tissue in an individual. This picture is a quite famous one. So even within the individual, in the skin, there are different aging, skin you can see. So I'm trying to explain about two different aging gap clock. The left one, mostly done by Yunte, one of my students. Brain MR image aging gap, I denote it as BAG. And right-hand side, you will see blood-based methylation aging gap, mostly done by Yono, the other student in my labs. So we can predict the MR images, and we can predict brain age using MR data, and we also can estimate AG from blood methylation data. So we estimate it, and we calculate the difference with the chronological age. If you see the plot here, it's quite well, high correlation you can observe. But what we see is that there are some differences. Some people have lower estimated age, and some accelerate age in this region. So we try to figure out what's the difference between this the high BAG or low BAG. So we gathered 3 different cohorts. We estimate the data, and we perform genome-wide association study with these BAG, the brain age gap. So we try to replicate whether those SNPs or genes associate with this BAG, and this is what we found. So the discovery phase, we identify 4 genome-wide significant SNPs and genes, and we try to replicate this to replicate 2 other cohorts, but we failed. But for each cohort, we recall BAG genes with a less stringent significance level, and then identify interacting particles or genes of BAG genes using gene expression data in brain tissue. And then we investigate the enriched GO, and this is what we found. These are the name of the 3 cohorts we used, and this is biological process enriched in interacting molecules and identified genes. So if you see this red color it's statistically significant gene ontology, and if you see the right-hand side here, it's the enriched geoterminology. And if you see that, and there are some exonogenesis and presynaptic functions are enriched. So if we identify BAG associated genes, and their functions are, synaptic functions or exogenesis functions. So it's quite, straightforward, but the boring answers we get. The other aging gap we measured is methylation aging gap, measured on blood. So if you see here, x-axis is chronological age, and y-axis indicate the methylation age gap. So if you see this plot, then there are slightly decreasing pattern across the age. So we divide the age group in 40s, 50s, and 60s, and 70s, and trying to identify whether there is some differences among the age group. And this is what we found. If you see this x-axis, it's the age group, 40s to 70s. And y-axis is, I just brought the biological process GO only. And if you see here, positive actin development adhesions are well enriched in 40 to 60s, but disappeared in 70s. And there are some other biological process, for example, muscle adaptation defense and immune receptor signaling pathways are not shown in 40 to 60s, but 70s, it's statistically significant. So we believe that, there are some AG group might have different genetic functions are captured by methylation age gap. So we try to go deeper look. As previous talk, the speaker introduced about the age-wise differences aging patterns. So we try to capture that. So we try to divide the age bin, but keeping the number of sample quite constantly. So we fixed as a 150 sample size for each bin, and we make it bin size might be vary, because if we want to keep the sample size constant, not to affected by the statistical effect. So keeping that sample size, the blue dot and line indicate the identified genes. So if you see here, such in 50s and late 50s and early 60s, there are more genes are identified, even though we're keeping the sample size remain same, almost the same. So we try to identify what's the genetic, the genetic functions are enriched in each bin. So I'm not going to tell you about the detail of this geo plot, but x-axis represent each age bin, and this is cellular compartment and molecular functions and biological process. And we will keep looking at the actin filament or cell, actin filament binding and active filament polarizations, and it was similar. So cell mobilities and adhesions are especially important in late stage. And there are some other genetic functions are enriched in particular age groups. So we try to gathering such information and interpret how it's related to aging. And we do have phenotype information is available for this data set. So we performed the phenotype association study, or PHAS. And I'm sorry about the masking there are some more signals, but I masked for clarification. But if you see this here, and spirometric, the lung functions are significantly correlated. We're still figuring out why those lung functions are such as high significant with MAG. So as you might already know, these are the hallmark of aging. There are many. So it's updated from 2023. And we believe that there are some upstream of these controlling this phenotype or hallmarks. In the previous work, we just look at the 70s because the data set was the maximum age was about 70s. But Professor Park and Colonel Lee, we have sequencing data for a centenarian, so we try to capture what will happen in centenarian. So we try to identify some fragments information with the extremely delayed aging group. So these works are performed by these 4 students. So we gathered longevity-related genes from a lot of genome-wide association study. So we identify longevity genes from publicly available data set with this keyword, and we try to see their functions. And we do have 111 centenarians whole genome sequencing data, so we try to calculate genetic risk of 61 disease trait using this amount of SNPs. And we are trying to interconnecting how these two are correlated each other or connections. So for the first one, we just captured the longevity genes and from many genome-wide association studies, and we do perform gene set enrichment analysis. And if you see this, cellular remodeling rate and immune aging are enriched gene ontology. And there are more, but I just show some, only this plot for your interest. And the other one is that we calculate polygenic risk score from GWAS catalog information. So we, as I said, we do have 111 centenarians genotype information. So we calculate PLS for centenarian, and we do have 4,000 just population. And so we compare those polygenic risk score with the populations. And if you see this, there are among 61, 17 disease and trait are different. So these 5 are just some examples. I brought only 5. So cataract and dementia, glaucoma, ovarian cancer, and skin cancer, type 2 diabetes. There are more genes. I mean, the phenotypes are statistically different among population and centenarian. And this is ongoing work. So we try to interconnecting among diseases using longevity enriched geo. So the 2 topics are different. So one is we just gathered information from GWAS data and gathered the gene, and then we identify enriched gene function. And the other one is that from the centenarian whole genome sequencing, we identify which disease or phenotypes are different from population, and we try to interconnect them. So these are the diseases different from centenarian and population, and we measure this thing with the gene ontology terminology. So these are the set size, and there are interconnections. So for example, for this case, diabetes and allergy is interconnected based on shared GO terminologies. And we try to identify what GO terms are hub things, so we perform network analysis. I'm not interpreting the result because it's ongoing, but I'm just trying to show you, as I said, some fragments. We are still gathering information. So many of the diseases are interconnected with some geotomes, and maybe if we find out some hub GOs, then it might provide some hypothesis. So that's what we are doing right now. So this is my end of my talk, but as I say, remaining things, I just provide some fragments ongoing in my lab, but I want you to build up some framework from the fragment. Thank you very much.
Thank you, Professor Kim. Let's move to the last presentation, plenary lecture by Professor Seo Yu-shin. I'll briefly introduce Professor Seo. She is a professor at Columbia University. I think I have to note that she is a director of reproduction, aging in obstetrics and gynecology. And she made a great contribution to epigenetic changes in aging, and so she received the Glenn Award. And today she will open a new field of ovarian aging. Yeah, please.
Thank you so much, Dr. Kim. That was a very nice introduction. I'd like to start by thanking Professor Sang-Chul Park and the other organizers for kind invitation to speak today. It's really particularly meaningful for me to be here because, as many of you may know, that I started my journey on aging research in Professor Park's laboratory more than 25 years ago. That's where I became interested in aging. And I remember every summer, Professor Park, with his multidisciplinary team, travel across Korea to study centenarians and collect blood samples. And so this is, that was the lab where I was introduced to centenarian studies, and I initiated my genetic studies of centenarians. And this very unique and formative experience really shaped my trajectory of career, and that's how I focus on my research. So today I'm going to talk about mechanisms of ovarian aging, and some of you may know that my lab has been taking human genetics and functional genomics approach to understand the fundamental mechanisms of aging and longevity. And this ovarian aging study is really very new. And I hope that by the end of this talk, I will convince you that by targeting ovarian aging, we may be able to make women live longer and healthier. That's the concept known as geroprotection. I mean, this audience is really well versed in aging biology, so I don't think I need to explain this, but I always like to start with this slide because I want to remind us how important it is to study aging in the first place. Brian and many other speakers already talked about this. We know by now that aging is the single largest risk factor for all chronic diseases, you name it, from osteoporosis to cardiovascular disease to different types of cancer, diabetes, eye disease, hearing loss, you name it, both men and women. So for example, if you're getting old, simply from age 50 to 75, increased risk of Alzheimer's disease by 100-fold, which really dwarf 10-fold risk of all known risk factors combined, which include ApoE genotype, being a female, hypertension, smoking, physical inactivity, and diabetes. And this trend is true for all chronic diseases, right? Cancer, kidney disease, cardiovascular disease, type 2 diabetes. So for those of us in aging biology, hypothesize that since aging is the main driver of all chronic diseases, by targeting the fundamental mechanism of aging, we'll be able to target many, if not all, diseases simultaneously. And this neuroscience hypothesis has been formulated based on many decades of very elegant studies done in model organisms, from yeast to worms and flies and mice, where we now know that aging is not just simple wear and tear. It's really regulated at the gene and pathway level. And by targeting those conserved genes and pathways, we can really extend lifespan and health span quite dramatically, at least in these preclinical models. And as Brian and other speakers introduced this morning, that we know a lot about aging biology by now, and it's referred to as hallmarks of aging or pillars of aging, so which include genomic instability, telomere attrition, loss of protein homeostasis, dysregulated nutrient sensing, signaling, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, etc. And the drug that target this fundamental process of aging, thereby extend lifespan and health span, at least in animal models, are collectively called geroprotectors. I didn't appreciate, Brian, that we know by now 1,000 such small molecules, which is quite staggering, right? So my lab has been interested in these breakthrough discoveries made in model organism to human system, asking questions, are these conserved genes and pathways really relevant to human aging and longevity as well? And more recently, though, we've been taking this reverse translational neuroscience approach, thanks to this huge, large data set from humans. So in fact, humans are by far the most phenotyped species in the world. We know a lot about very detailed pathology when it comes to age-related diseases and pathology, right? So combined with this large-scale data set, including human genetics, epigenome, together with electronic medical records, we can really use human as a discovery platform to generate hypotheses and test them in human cell and animal models, of course, and we can translate back to clinic, right? So today, my data that I'm going to talk about, this ovarian aging, is really one of the best examples that I can think of in terms of reverse translational geroscience approach. Why do we care about aging in women then? First of all, historically, there has been inequity in biomedical research and clinical development. You may not appreciate, but far majority of basic research has been done in male animals and male subjects. It's not just humans, and even flies was focused on male flies. So there's a huge imbalance. To the point where in, I think NIH, 80% of NIH-funded research just focus on male animals. So in 2016, NIH mandated inclusion of both sexes in research design and analysis and report. It's called sex as biological variables policy. And we know by now that the sex differences in aging is huge. Okay. Combination with insufficient funding, I should say, lack of funding. This is example from investment bank. And you can see that less than 5% of the total NIH funding is dedicated to women's health. Actually, other than gynecological cancer, it's really less than 1%. Okay? You cannot discover what you don't study. So we know very little about how female system works, let alone age, right? And this lack of knowledge or cost of not knowing has enormously profound negative impact on women's health, because women are likely to be underdiagnosed, misdiagnosed, or diagnosed later. They suffer from more drug side effect, or the treatment is less efficient in women because they were never part of clinical trials. Thankfully, times are changing, and there's increasing awareness and public attention calling for change. And end of last year, actually, Biden administration initiated Women's Health Initiative, and NIH embraced the concept and started to publicize this notice of special interest in women's health research. But under current administration, we don't know the fate of this special interest yet. So finally, why do we care about aging in the ovary? You may not really have not appreciated this, but ovary is really the very first organ to age in the human body. Its function declines in your mid-30s with rather rapid decline and completely shuts down when women enters menopause, okay? And if you look at functional decline in other organ systems, aging in the ovaries really stood out as premature, right? So ovaries in a woman's body already in geriatric state in their 30s, whereas the other functions are at its peak performance. So there's a huge gap among other organ decline and ovarian function decline with age. And as you know, that in the last 150 years, the average life expectancy has increased or doubled. But interestingly, the age at natural menopause stays largely unchanged. That what does that mean is that more and more women live ever greater proportion of their lives in postmenopausal state. And we know it's really bad because menopause coincides with very cascade of very deleterious health outcomes in cognitive, immune, bone, and cardiovascular system, among others. And in fact, I think Brian showed this morning that menopause accelerates biological aging as measured by epigenetic clock. In fact, this Levine and Horvath study showed that there's a genetic component of this menopause-induced biological aging. And consistent with this genetic link, women with later menopause live longer than women with the earlier menopause. And very intriguingly, brothers of women with later menopause has pro-longevity benefit than brothers of women with earlier menopause. So in other words, if we understand the mechanism of menopause or ovarian aging, we'll be able to benefit not just women but also men. I will come back to that point. So this data suggests to us that ovary does much more than reproduction. It influences overall health in women. So what happens if you remove ovary? Okay, so ovariectomy or oophorectomy increases risk of not just single diseases such as dementia, cardiovascular disease, type 2 diabetes, but it also increases risk for multimorbidity and all-cause mortality. Again, indicating that ovary has much more than reproductive function, influence overall health in women. Finally, in mice, just simply replacing old ovary from old mice with a young ovary from young mice by transplantation is enough to extend the remaining lifespan and health span by improving insulin sensitivity and reducing inflammation. Okay. So as a geroscientist who trained in biology of aging, it was crystal clear that we have to target biological mechanisms of ovarian aging for geroprotection in women. But as I said, through this historical inequity in biomedical research, we know very, very little about mechanisms of ovarian aging. What's relatively well established, though, at least in humans, is this huge variability in rate of ovarian aging as measured by age and natural menopause. So this is the data from UK Biobank, and you can see that huge variability, right? Some women undergo menopause in their 30s, some women dip in the late 50s. Now you can take this individual variability in onset of menopause as a trait and can perform large-scale genome association studies. And one of the most recent study, which involved huge number of women, more than 200,000 individuals, identify 290 loci that are associated with age at natural menopause, or timing of menopause. And when we look at these genetic variants, single nucleotide polymorphisms, 94% of them occur in the non-coding region of the genome. So they don't have anything to do with the protein coding region, suggesting that if anything, they contribute to this inter-individual variability by gene regulatory changes. This poses huge problems because human genome has very extensive linkage disequilibrium. So in a single locus, you find many, if not 100, linked genetic variants. That means you have no idea which is causal variants that cause these regulatory changes. And regulatory elements, especially enhancers, regulate genes from long distance through this chromatin looping, skipping many genes in between. So to identify causal genes that are under influence of this causal variant is a priori and unknown. And you know that many regulatory elements, especially promoters and enhancers, they have cell type specific functions. So cell types, causal cell types that are under influence of this variant and genes are not known, let alone mechanisms. So what we and others do is to take genome-wide association study data and integrate them with epigenome and epigenetic data set collected from humans, identify those variants that occur in regulatory elements, especially transcription factor binding sites, and try to predict causal genes by looking at the EQTL association. This is expression quantitative trait loci correlation, meaning that you are correlating genotype with changes in gene expression, and of course, there's chromatin interactions. And for this type of integrative analysis, CATLAS has been our Bible. It has single cell ataxic data from 220 cell types of over 30 human tissues. But there was no data from ovary when we started this work, which is really unfortunate because ovary is a very complex organ with many different cell types that maintain and support ovarian function. Shown here are 8 major cell types in the ovary. So for example, theca cells and granulosa cells are 2 main components of follicles that maintain oocyte growth and development. So these are ovary-specific cell types. But you can also see other cell types like stroma cells, epithelial cells, smooth muscle cell, endothelial cells of blood and lymphatic origin, and immune cells. Since we know very little about mechanisms of ovarian aging and we don't know how to interpret these GWAS, non-coding variants, Chen Jin, a postdoc in my lab, decided to take more unbiased, comprehensive, systematic analysis by using single nucleus multi-omic analysis. So what Chen did was take ovary from young women in their 20s and ovaries from reproductively old women in their early 50s, late 40s, early 50s. So we are now talking about aging from 20 to 50s, and performed both single-nuke RNA-seq analysis for aging transcriptome across different cell types, as well as single-nuke ATAC-seq analysis for regulatory changes. With this data, we can now integrate the GWAS with the epigenome or the regulatory information we generated. For example, from ATAC-seq data, we can assign variants that occur in regulatory elements that are active in ovarian cell type. And we were quite surprised to see that those variants actually are shared across many, if not all, cell types in the ovary. This means that the effect of variant can be quite global, at least in the ovary. When we look at putative target genes, so one of the problem, in my opinion, when people interpret GWAS, they take a nearest gene as a target gene without knowing the real causal genes, okay, which is not always true. And nevertheless, using that approach, we ask questions: where do these genes that are marked by variants are expressed in ovarian tissue cell types? And then we found them actually, they are enriched in genes across all cell types in the ovary, which is huge contrast to GWAS from polycystic ovary syndrome and ovarian epithelial cancer, which is derived from granulosa cells for PCOS, or epithelial cells for ovarian epithelial cancer. Again, this indicates that menopause-associated genetic variants have a global impact on the gene regulatory network in the ovary across different cell types. Using this data, we can now start looking in detail what are the causal variant and causal genes. I will give you one example. So in this case, in the Helby locus, we found variants that's associated with later menopause, which occur in the regulatory elements that is active across all cell types in the ovary. Interestingly, when we look at the expression correlation between this genotype and the later menopause associated variants, it's correlated with the reduced expression of Helby gene, okay, in the ovary. Okay. Interestingly, though, when we look at the association across all human tissue type, it was not just ovary. Every tissue type that have been profiled in GTEx database show correlation. Ovary is not even the largest effect size. Look at this muscle has the largest effect size. So, and this correlation occurs not only just women, but also men, right? Again, this may be the link, how this menopause associated variants may influence male siblings' pro-longevity outcome. And this variant is predicted to bind less efficiently to transcription factors. So what I have shown you is that we take this larger scale genetic data, integrate them with epigenome data to identify, to predict causal genes and variants in cell type. And of course, this is prediction and at most correlation. The question is, how do we bring this genetic association to causality by establishing experimental validation? We've been really thinking hard and long about this problem. It's not easy. But what we have decided to do, which is spearheaded by another postdoc, Jiping Yang, to take human pluripotent stem cell, both embryonic stem cells and induced pluripotent stem cells, and engineer them with CRISPR so that the cell carries one particular predicted causal variant. And then using that engineered stem cells, we differentiate them into multiple cell types of ovary, including granulosa, endothelial, smooth muscle, and stroma cells. And using these isogenic cell types that differ just by one single nucleotide change, you can really confirm, experimentally validate regulatory output by looking, is this really causal variants? Is this causal genes? And is this cell type that are influenced by this gene and variants? And most importantly, what are the directional impact, right? So is this variant make your gene express more or less? And then using this two system, you can really deeply drill down the mechanisms, including transcription factors that's involved. I'll give you one example. So again, this is B locus. For the regulatory variants, the best approach is to take heterozygote cells because, as I said, there are many variants that are linked, right? So you take heterozygote cell that carries the variants as one associated with later menopause, one reference allele, and then you identify linked variants in the coding region, and then you can look at the relative abundance of mRNA, which is measured by cDNA, of course, as a relative regulatory output, right? So here's the example. First, control experiment. When you take genomic DNA, of course, it's one copy versus one copy. You have 50 versus 50 contribution when you perform allele-specific PCR or allele-specific expression analysis. But once you take cDNA from the differentiated cell type and perform allele-specific PCR, then you can clearly see now the later menopause associated variant now cause reduced expression of Helby, not just one cell type, but across all cell type that we have tested, which was predicted from EQTL analysis. We're also lucky to get primary granulosa cells from IVF clinic, heterozygote carrier for donors, same thing. the later menopause associated allele cause reduced expression of HELB. The HELB is a helicase B gene. It's an inhibitor of accurate double-strand break repair homologous recombination. So when you reduce its expression or function, you predict that you have increased the accuracy of DNA repair. So in collaboration with Matt Yousefzad at Columbia, we have generated HELB knockout mice to mimic the later longevity variant effect, which reduces expression of HELB. So using this mouse model, we are going to test, of course, is it going to improve reproductive longevity, number one. Number two, does it really improve health span? Because that's our premise, right? The aging in the ovary, if you delay it, may promote health span in women. So this is going to be tested. I have this wonderful postdoc in the lab, Shweta Dipali. She, unlike me, she's a hardcore reproductive scientist trained in Francesca Duncan's lab in Northwestern. So I look forward to this data. I told you about just one example, HELB, one variant, okay. When we performed this in silico annotation analysis, we found over 100 candidate regulatory causal variants. And how are we going to test this? I mean, some of you may have experienced this CRISPR and stem cell engineering. It's really very tedious, especially when you have to differentiate them into multiple cell types, right? So we need a high throughput, high to medium throughput screening tool or approach to identify and confirm and validate causal variant genes in endogenous genomic context, as we have seen in CRISPR engineering. Jiping, again, spearheading this project, he decided to take CRISPR interference screen combined with a single-cell transcriptome readout. So you can see it in this hypothetical situation that here's the putative regulatory variants that occur in cis regulatory elements, gene-rich region, which is always the case in the human genome. And as I said, GWAS literature, when you see it, they pick nearest gene as their target gene. It's not always the case. So how do you confirm that and identify causal gene? So by using this dead Cas9 repressor targeting these variants, then you see this, oh, now it's gene F is the true target gene of these variants, right? And then by performing transcriptome-wide analysis, you can really look for regulatory output globally in different cell types. So Jiping designed very elegant CRISPR-I, single-cell CRISPR-I method. So he generate guide RNA libraries. So each variant, he generate at least 5 guide RNAs that's compatible to 10x platform, and use very strong repressor that Cas9 fused with CREP and MSAP2, which is, in our hands, it's much more potent than CREP alone, and perform this single-cell CRISPR-I screen of the all 100 variants that we predict to be causal. The assay worked beautifully. This is the data from granulosa cells. And then you can see about 70 genes that are affected by this screen, and majority of them show reduced expression, as you would expect from interference of CRISPR-I. These are the genes that we have detected, and the size of the gene name indicate the frequency of the detection, and you'd really appreciate that function of this gene have not been really well characterized. Many of them, we don't know what the functions are. But many of them are implicated in key aging pathways, for example, including HELB, genome maintenance, like NRBP1 and zinc finger 638. That's relatively new study, came out in preprint in bioRxiv. Protein homeostasis involved genes and genes involved in mitochondrial function. And one of the genes that we are following up is, before I talk about this, I'm going to show the real results. We are very happy to see our causal variant in HELB locus that show reduced expression of HELB gene that associate with later menopause. When we target this, not the neighboring genes, it really influences HELB. So we sort of confirm our the individual validation data. What are other genes? This causal variant, when interfered, that it downregulates a gene called the CPNE1. Okay, and you can appreciate huge significance, and the downregulation was a lot, more about twofold. Interestingly, the CPNE1 variant is predicted to change binding of transcription factor called the CTCF. And indeed, in this locus, you see active regulatory elements, it occurs. So you can predict, at least in the ovary, the CPNE1 variants influence binding of CTCF, thereby influence expression of CPNE1 in ovary cell types. This was very exciting results. When we look at GTEx database, this CPNE1 expression is highly prevalent in reproductive tissues, including uterus, cervix, and ovarian fallopian tube. So clearly, it might have ovary or reproductive tissue-specific activities. Nothing much known about this gene function, and we are actively studying those. But what interests us most is when we look at the trans effect, meaning the broad global impact on transcriptome. Targeting this CPNE1 variant has largest changes in the target gene. There are like 700 genes. It changes in expression when you did CRISPR screen. About 40% of those are influenced by CPNE1. And when we look at those differentially expressed genes by inhibition of a CPNE1 variant, those are the usual hallmarks of aging, including apoptosis signaling, reactive oxygen species response, oxidative phosphorylation, chromatin organization, ribosomal biogenesis, and so on. We are actively studying the function of CPNE1, and we are also setting up this CRISPR screen, starting with granulosa cells, but we are also doing epithelial cells, stroma cells, and fibroblasts. What about aging transcriptome? When we look at the single cell, single nuke RNA-seq data, two things surprise us. Number one, as you may appreciate, that among 3,000 genes going up and down with age, that they show the same directional changes across all cell types in the ovary. So this high level of coordination was quite interesting. And we wanted to study those a little deeper. And you can look at the pairwise similarity through the analysis, and you see that there are really a high level of coordination among cell types, different cell types in the ovary. And the reviewer asked us, is this common phenomenon in aging in different tissues, or this is ovary specific? So we had to look at other tissues in human body, major tissues including brain, liver, and reproductive tissues such as breast. And you can see that the level of coordination is not even close. So this huge level of coordinated transcriptome change across cell types in the ovary is quite unique. Number two, when you look at these differentially expressed genes, they are enriched in so-called hallmarks of aging, right? So here's some examples, right? So here, mitochondrial dysfunction, deregulated nutrient sensing signaling, loss of protein homeostasis, epigenetic alteration, genome instability, senescence, telomere attrition, and so on. But what caught our eyes was this upregulation of mTOR signaling pathway across many cell types, if not all cell types, in the ovary, right? So again, using that other human tissues that we study this coordination, we look at the tissue level implications by comparing amplitude index and prevalence ratio. And of course, compared to other tissues, ovary is really specific, showing this dramatic upregulation of mTOR signaling. And many of you know that mTOR signaling is the central mechanism of aging, and its inhibitor, rapamycin, extends lifespan and healthspan in all modern organisms that have been tested thus far. So it's one of the most robust geroprotectors that we know of. This study showing that mTOR signaling is upregulated across many cell types in the ovary suggests to us that ovary can be a geroprotective target of rapamycin. In fact, in preclinical studies in mice and rats, rapamycin treatment increase ovarian lifespan and fertility, increase ovarian reserve, preserve ovarian follicular pull. So together with our study, we really were confident that rapamycin will improve or delay aging in the ovary. So we started clinical trials together with Zev Williams, who's the director of Columbia Fertility Clinic, to test the effect of rapamycin in improving reproductive lifespan in women. And the trial is called Vibrant, validating benefits of rapamycin for reproductive aging treatment. It's a small proof of concept study. We are using very low dose of rapamycin, treat women in their 35 to 45, 35 when ovarian function started to decline, for 3 months. And then we are following them up for additional 9 months, okay? We finished all 50 people, of course, half placebo. Very encouragingly, it's very safe. No side effect, no toxicity, even it does not influence menstrual cycle, okay? And the primary outcome is ovarian reserve, and we're going to do multi-omic analysis for biomarkers and mechanistic insights. It's still blinded, so I think the study will be finished before the end of this year. But you would expect that maybe ovarian reserve, diverge in 2 patterns. Actually, that's what we saw. It's quite encouraging. Based on this result, we started a multi-center trial. It's called the Vibrant 2 study. In fact, I submitted this grant 2 days ago when I was in Korea. So these are schools that are involved in this multi-center trial, and this is a little bit longer treatment for 9 months. Initial Vibrant 1 study, we wanted to do 6-month treatment, but IRB said no. So we had to reduce it to 3, but in this case, a 9-month treatment for additional follow-up. So this is a 15-month study. We'll be very enthusiastic to see the results from Vibrant 1 and 2 studies. And obviously, human clinical trial is absolutely important and needed, but it's limited. So for example, we cannot get their ovaries to see what happens in the ovary, right? So we decided to recapitulate human trial in mouse study. so that we can have mechanistic insights using many different tools, including single-cell multi-ohm studies. This is by far the largest project that we have ever done in my lab, involving many people in the lab. And we just finished single-cell RNA-seq analysis. We are performing now the epigenetic clock analysis collaboration with Vadim Gladyshev. So hopefully, maybe a year from now, I'll be able to talk about the results. So to summarize my talk, I hope that I convince you that ovary is not only important for reproductive health and lifespan, but also overall health in women. Okay? And by integrating human genome-wide association studies with epigenome data, including our own single nuke, ATAC-seq and RNA-seq, we identify or predict potentially causal regulatory variants and causal genes and causal cell type, and validate them using CRISPR stem cell engineering followed by differentiation model. Through the single nuke RNA-seq analysis, we have shown that aging in the ovary is a true accelerated, true model of accelerated aging. And those proceed with the known hallmarks of aging. So what that means is that the known geroprotectors that have been tested in animal models can be used to delay aging in the ovary. So we started the Vibrant study as a proof of principle using rapamycin as an example. Hopefully, we'll increase the repertoire of drugs. I was once interviewed by a reporter in Business Insider, so I pitched to her. The other flip side of this is that ovary may be a best way to translate preclinical tests to humans, because when you're testing longevity drug or geroprotectors in humans, it will take years, if not 10, 20, 30 years, right? And she was brilliant. This was her title, a radical new idea to speed up anti-aging research, test ovaries, not mice, right? So ovary can be really used as a fast testing system for geroprotectors. With that, I'd like to thank members of the lab. I think I acknowledged most of them. Chen and Jiping really single-handedly led this project together with others. And I'd like to thank wonderful collaborators. Without them, this project would have been not possible. And funding agencies, especially NIA. Okay, I'll stop here. Thank you for your attention.
Thank you, Professor Sir. Well, we'll have a discussion session. Panelists will come up.
For discussion, all of speakers from come here and sit the chairs on the front. And panel discussion members, please sit here. No. Speakers, yeah, because speakers have to be answered about your questions. Yeah, first of all, we will receive some questions from the panels, and then we will open the floor.
Can I ask a question first? Yeah. Thank you all for the nice lectures, and I'm Obin from Seoul University. I have a question to Professor So. So you showed something interesting that the ovary shows mTOR signaling changes that go into the opposite direction from the other organs. So my question is how can we target the ovary specifically without causing any other aging effects in other organs?
Yeah, so that's an interesting question. I would like to clarify the direction is not opposite. So what's happening in other parts of the body when you are in 70s and 80s occur in the ovary in your 30s and 40s and 50s. So it occurs in such an accelerated way, but with the same mechanisms that we already know of, so-called hallmarks of aging. So that's what we are saying. Take the geroprotectors that target those hallmarks of aging, test it in ovary. If it works, okay, you will improve ovarian function, delay aging in the ovary, thereby improve women's health. But at the end of the day, they will help overall health. Okay. But thank you for that question.
Okay, I'm Kyung-Chul Yoon, Jeonnam National University. I have a question to Professor.
난 영어로 안 하고 한국말로 하겠는데요.
I have a question to Professor Park Tae-jun. Okay, thank you for your nice study and work in the skin, and you focus on the mid-old cells in the pathogenesis of skin age. I agree with the role of mid-old cells in fibroblast, and elimination of senescence fibroblast may help to delay the skin age. How about the role of the immune cells? You know, as people age, many defects in the immune system and increase in the inflammatory cells and CD4 T cell, B cells, and decrease of regulatory T cells. So there is also the importance of the immune system. And how do you think about that?
Thank you for your very interesting question. We also tested the immune cell senescence in old-aged tissue. The senescence T cell secrete the coenzyme A and B. Also, and this coenzyme can induce the PAS signaling in epithelial cell, and then can induce the epithelial cell apoptosis. And I thought that the immune cell senescence can regulate the epithelial function via the regulate the apoptosis. I think. Thank you for your very interesting question.
Sorry, Dae-hee Hwang from SNU. I have a specific question first, small specific question to Professor Seo. And when you test the causal link of the identified SMP to the expression of HER-B, you use the differentiated cell, right? I wonder whether you checked these differentiated cells are sufficiently similar to those the corresponding cell types in the endogenous setting. So by performing the bulk RNA sequencing or single RNA sequencing, that's my question. First, a small question.
Yeah, thank you. Those studies have not been done. So, but when people establish differentiation protocol for ovarian cell types, not all, but many did that homework. So for example, granulosa cell differentiation protocol was established in George Church's lab at Harvard, and he used different ways, including transcriptome, to see what are the most similar to in vivo changes.
Okay. Other people already confirmed the protocol, that's what you're saying?
Well, other than granulosa, the stroma cells and endothelial, they are like generic differentiation protocol. But when it comes to ovarian cell type, we don't know, right?
Okay, the second question. So from my experience, the translation is getting decreased actually as the many biological system age. So by treating mTOR rapamycin, so we can expect that translation activity will be improved, right? So I thought like a benefit is kind of expected already.
But first of all, rapamycin does much more than just
Yeah, that's true.
So I think collectively the beneficial effect is large. Maybe Brian can have a little bit of comments because this was addressed a while back, right? Brian, do you want to mention something? Yeah, to clarify.
So we don't know in the ovary, but we looked in a couple other tissues many years ago, and what you find with rapamycin is it doesn't alter translation levels. What it does is it changes translation levels. So if you acutely treat an animal with rapamycin, it will shut down translation. But if you continue to treat the animal for a month and then look after the last treatment, you will find no change in general levels of translation. But probably through 4-EBP1, which was on your list, you get changes in specific messages. And so I think that's the bigger long-term effect of rapamycin.
Then see, this is a whole body treatment or this is treatment specific to ovary?
Oh, no. This is you take a pill, low dose, once a week. So the big question is, what's the right dose?
So
right?
effect on the ovary actually can be indirect, like the whatever drug you treat.
That is
it
true.
affect brain first and then maybe
That is true.
brain
Yeah.
indirectly affect
Yeah.
ovary. How can you distinguish this direct and indirect effect?
What we have to remember, in that age, 35 to 45, mTOR is not active in other tissues, unlike ovary, which is very high, right? So if anything, the target in the ovary makes sense because it's activated with aging and in pathological conditions, right? Oh, I forgot to mention, and it's really interesting in mice, when animals treated with rapamycin, the effect of lifespan extension is far larger in female mice than in male mice. So we like to hypothesize that ovary mediate the stronger effect, but we don't know. So, we are talking about, why don't we do ovaryectomized and then treat with rapamycin, and does this sex difference disappear, right? So this is something that we are interested in testing.
For your last comment, is the longevity especially in the female mice, dependent on the estrogen or the sex hormone signaling or not?
I don't think anybody has tested that, but what has been tested, which is really very interesting, obviously hormone is number one candidate, right? So I explained this transplantation experiment, right? You can take a young ovary and then put it in the old mice. When it's a small study, but the result is really very interesting. So what they have done is that before they take the young ovary, they use this chemical to remove follicles which produce sex hormone. So now you have ovary with only soma. And then, what happened in the old, but that was still enough to extend lifespan. So in other words, the soma of ovary has pro-longevity effects in old animals.
So when you start treat the rapamycin during the course of lifespan, does it matter or it doesn't matter?
Were you a reviewer of our paper? No, just kidding. That's a very important question, which we haven't addressed, because our mouse, the single cell studies is done between young and, not humans, sorry, young and old, right? Young and reproductively old, 20 and 50s. Okay. The question is, what about in between? Because you said, ovary gets old already in your 30s. So when should we take it, right? So do we see the signature of mTOR already in your 30s? We haven't tested that, okay? But the reason why we target those 35-year-old women is because the functional decline is already pronounced in the mid-30s. That's why we decided to do that, right? This is a study that we have to do.
Okay. Oh yeah. Hi. This is Sajung. I have a question to Professor Kwang Song Park. So, as you know, recently the AI, artificial intelligence, is a hot topic, and the government is about to pour in $100 billion. And for the elderly and for the longevity, would there be any possible good applications of the AI for the like the centenarians and for the healthy aging?
I think Professor Sajung Yoon can address about because you are pioneer to use the AI in the field, and but I expect that actually you can use AI because it's based on the big data, and then we have very small data in centenarian. We need more data, but possibly we can use AI for predicting something in our old age. But you also can comment on your question.
I think that's a great answer. Thank you. And I have another question to the Professor Chung-Soo Kim. So I was listening to Dr. Seo's presentation, HLB, for the ovarian aging HLB expression, which is DNA helicase B, NRBP1, which is the nuclear receptor binding protein, and CPNE1, which encodes calcium-dependent membrane binding protein, etc. And I was thinking that nowadays we are talking about the aging as a more like a Newtonian mechanics. But if you think of Einstein's relativity theory, then it just gives me the insight that maybe the dimensionality might bend, comparable to the mass. So in this case, I'm thinking of the absolute biological clock. So is there any good insight could you share about the relative, instead of the regular Newtonian mechanics versus relativity of the aging?
Well, that's a tough question. So if I recall your question correctly, then you were asking about, can I give some relativity aspect of idea to instead of Newtonian?
Yeah, Newtonian mechanics basically based on the mass and all these things. But Einstein was thinking about the dimensional bending, right? And I think there could be some parallel interpretations be possible.
So the key point of the relativity of Einstein is that general relativity is about space-time incorporation. So three dimensions in space and one dimensions in time can be parallelly, you can coordinate them in one axis in vector, and then time frame, the four-dimension frame remains with the velocity as a constant. So it's not easy for me to answer that question. Maybe he's asking that question because I majored theoretical particle physics in my my graduate. So, but well, it's a difficult question. But I'll figure out and I will let you know how I can provide that answer later.
Now we will open the question on the floor. So if you have any questions, please.
좀 서유진 교수님한테 여쭤보고 싶은데 제가 지금 질문 자체가 좀 잘못됐을런지도 모르겠는데요. 두 가지 한번 여쭤보겠습니다. 첫 번째는 라파신을 쓰는 경우하고 이 에스트로겐이나 프로게스테론을 쓰는 경우하고 차이점이 뭐인가? 두 번째는 여성 에이징 프로세스는 갑자기 오는데 남자에 비해서 여자는 왜 갑자기 오느냐? 그걸 해결하면 되지 않겠느냐 하는 문제에 대해서 좀 얘기 좀 해주셔서 감사하겠습니다.
Can I speak in English or should I say in Korean? Yeah. So the first question, which is very important question, was what's the benefit of using rapamycin compared to the generic hormone replacement therapy like estrogen? I must say that estrogen therapy is like a band-aid. You already are in the track. It's already happened, and you try to treat symptoms. You're not addressing the root cause. Okay, whereas rapamycin is targeting the fundamental processes, not as a consequence. So that's a huge difference, right? So hormone, for example, is not, the hormone treatment is not going to extend reproductive lifespan. That's well established. Okay. And the second question, sorry, I forgot what was the second. Oh, right. Yes. So there are wonderful minds hypothesizing or theorizing from evolutionary point of view why menopause occur in such a rapid way in the ovary, right? Actually, menopause is typically humans. It's not evitable. Only humans and a couple of whales undergo menopause. So people are thinking maybe we can completely get rid of menopause, which I don't think it's going to happen. So there's a major hypothesis. It's called grandmother hypothesis. So during evolution, when your resources are limited, rather than competing with your own daughter, you shut down your capacity to reproduce. Instead, you take care of your grandkids. Thereby, you have increased chances for spreading your genes. And actually, in Africa, in hunter-gatherer tribe, there is a study showing that there's a correlation between the presence of grandmother with the well-being of the grandkids. So that's one of the major theories why, as if it's programmed, that the shutdown of ovary function occurs in such young age, right? So it's the realm of evolutionary biology, and I must say that that's not my forte, but that's major hypothesis.
Okay, last question.
Yeah. I'm Konori from Joseon University. I have a question to Dr. Seo. As you emphasize the menopause is critical for the woman's aging. To my knowledge, generally the average lifespan of woman is longer than the man. So there is some discrepancy between your hypothesis and the real world.
Yeah.
Could you explain that?
So that's an interesting question. So 2 points. Number one, you are absolutely right. I think Dr. Park also showed the data that anywhere in the world, women live longer. It doesn't have to be centenarians. It's just, even if your life expectancy is 50s, like Nigeria, versus Japan, the average life expectancy is 86, they live longer. Okay, so women outlive men. That's clear. And in terms of mechanism, we don't know, okay? But what's well established is that while women are living longer than men, they are in general sicker than men. So they have a higher physical inactivity, disability, more days of hospitalization. So women are less healthy in general. So in my opinion, women are resilient to death, but they are not necessarily healthier. Okay. The other thing that I really want to clarify people think that menopause is ovarian aging. No. Menopause is a consequence of ovarian aging. The ovarian aging occurs 15, 20 years before you hit menopause, right? And that's just marking the end of ovarian aging. So those are two things that we have to really differentiate.
Yeah, I have one simple question, Professor Yuri Kim. Your diagnostic tools for blood pressures is very important in elder people. So, how can we? What's your opinion? How can we, if it is possible to monitoring of some specific age of a whole age your monitoring systems as a biomarkers of aging process? Is it possible to monitoring in measuring of biological age in human?
You mean the continuous monitoring of the ECG or the blood pressure can be the biomarker to the aging process?
Yeah, during 1 week or 2 weeks.
Yeah, it is possible because some patients has their arrhythmia in younger age, but the other patients is over 90 years. They got their first AFib episode in that time, so there is quite difference from those two persons. Like an ovary, the 30 years the menopause and the 50 years menopause at the same time in the heart. Well, so the continuous monitoring can help to distinguish those two kinds of groups, but not now. So we get a bit more data and more analysis for that. So maybe it is possible to the tool for the aging process.
Actually, can I make one comment? I know that in Korea, there's a huge interest in senescence and senolytics. And as we all appreciate, there are two characteristics of senescent cells. They are extremely rare, so it's very difficult to detect, and they are very, very heterogeneous. So the inducers of inducing signals of senescence manifest in different ways in cells. So it's a huge issue. How do you know that your senolytic is going to work? So NIH has initiated a senescence mapping center consortium. This is funded by Common Fund. It's called SENNET. So both mouse and humans, we are mapping across lifespan from 20s to all the way to 90s to identify, first of all, what are the cell type specific senescence markers, because we don't know, and how they occur and how they influence neighboring cells. So I really want to encourage you to look at the database, the SENNET database, and it's really treasure trove, right? So there's a single cell and spatial transcriptomic. It's a beautiful data set, so I encourage you to utilize that data for your own research. Yes, both human and mice. So this is NIH Common Fund. They don't fund mouse study, but the reason why they did it was to measure a senolytic effect. Okay. It's a beautiful database. You should really look into it.
Thank you for precious information. Yeah, I'm very sorry stopping here this morning session. Yeah, thank you all the participants, speakers, and panelists. Thank you very much.